to those who are following us from other time zones. Welcome to today's workshop. It's my pleasure to welcome you to the Montreal Neurological Institute-Hospital. We refer to it as the Neuro, and you will be hearing me and other speakers today use the Neuro as our reference to our beloved institution. Today we are going to hear a number of speakers that will discuss how here at the Neuro we have been implementing open science practices to advance and promote the pace of discovery and treatment in neurological diseases and disorders. My name is Stefano Stifani. I'm the Associate Director of Research here, and it is my great pleasure again to welcome all of you to today's proceedings. Now, as I'm sure most of you already know, today's workshop is actually part of a more elaborate and rich series of events which have been put together by the Fondazione Prada, the Prada Foundation, indicated here, which is headquartered in Milan, Italy, as we'll tell you more in just a second. And this is a component of a larger initiative called, and just advance, called the Human Brains and Preserving the Brain Initiative. And so let me tell you a little bit about how the Prada Foundation has been a leader in supporting advances, awareness, and in general, knowledge in the field of neuroscience, and particularly neurological diseases and disorders. So today's workshop is part of this large initiative that started already years ago, the Human Brains. And in particular, this year in 2022, there are three main components to this initiative. One that I will just tell you in a moment is the forum, it's referred to as the forum on neurodegenerative diseases, which is taking place already in Milan, Italy. And then a series of workshops like the one today, which are also part of this initiative. So let me start by telling you a little bit about the forum. Sorry about that. I want to go next. So the Preserving the Brain initiative is today, this year, is the fourth phase of a project started in 2020. It is centered this year around the forum taking place in Milan, which is comprised of two main events: an exhibition running from September 16th to October 10th, so already ongoing. And for those of you who work at the Neuro, I'm happy to tell you that the Neuro has an exhibition at this event. And then a conference, a two-day conference on the 6th and 7th of October, coming up soon, and the Fondazione Prada headquarters in Milan. And the forum participants will include researchers, patients, associations, will include representatives from pharmaceutical and biotechnology industries, as well as policy advisors. And the main aim really is to debate and discuss together the current state of knowledge in the field of neurodegenerative diseases and the tools that are currently used to fight these diseases. This event is open, will be streamlined, so anyone who's interested will be able to follow the proceedings. And of course, anyone who is in Milan will be able to attend the exhibition, which is open to the public. The Fondazione Prada has worked together with 13 renowned institutions around the world to put together the program of this initiative. These are indicated here, and I'm very happy and proud to say that the Montreal Neurological Institute-Hospital is one of these 13 institutions that have worked together with the Prada Foundation to assemble the program for these events. One thing that the collaborating institutions do, in addition to contributing to the program, is to hold a series of local workshops. So this is one example of such local workshops, which are held by the collaborating institutions. So this is the third component of the Human Brains Preserving the Brain initiative. And each workshop will focus on a different theme. Today, as you know, we will be hearing about science and other workshops will address other themes, but they all have the main objectives of discussing advances in new treatments for neurodegenerative diseases. These are all available online, so in addition to participating in today's workshop, of course, everyone is welcome to attend and contribute to other workshops as well. So I want to say thank you very, very much to the Fondazione Prada for putting together this exciting program, and in particular to Professor Giancarlo Comi, who is the president of the scientific board of the Prada Foundation of this forum on neurodegenerative diseases. So now on to today's presentation. So some of you already know by being here at the Neuro, at the Montreal Neurological Institute-Hospital, that we have invested considerable attention and considerable resources in developing open science practices in order to accelerate not only scientific discovery, but also the development of new treatments and delivery of cures. And I want to briefly tell you a little bit about how the Neuro has become a leader in open science practices. As early as 2016, the Neuro decided that they were going to become fully committed to the implementation of open science practices. And following an 18-month buy-in process, we adopted guiding principles for the implementation of open science. And then we were the lucky recipient of a very generous donation from the Annabel Foundation, $20 million, to start the Larry and Judy Tannenbaum Family Foundation, Tannenbaum Open Science Institute, TOSI, as we refer to it. And you will hear later at the end of this talk from the Chief Executive Officer of the TOSI. The Tannenbaum Open Science Institute leads the implementation of open science within the Neuro. It also encourages other Canadian neuroscience institutes to commit to open science. And it also goes beyond the Canadian borders to really try to trigger a global conversation about the importance of open science and promoting the spreading of open science practices. And again, open science practices are not only limited to what most people usually think about, things like open science publishing, or open access publishing, I should say, but in fact, include much more than that, include the development of openly accessible biorepositories, the free sharing of data, the promotion of early drug discovery through open science, the distribution of findings without borders and barriers and delays. And today we will hear about some of these very, very important practices. So before we start, let me thank the four speakers today who will present in these workshops. We will hear first from Professor Jason Karamchandani, who is a neuropathologist and who will tell us about the extremely exciting clinical biospecimen imaging and genetic platform that we have developed at the Neuro under open science modalities. We will then hear from Professor Thomas Durcan, who is the director of the Open Science Neuro Early Drug Discovery Unit. And we will learn how open science is promoting discovery through collaboration and outreach. We will then hear from Professor Joanne Stratton, who will tell us about open science is helping us to understand Parkinson's disease, immunological mechanisms better. And last but not least, we will hear from Professor Sali Farhan, who will tell us about open science is helping us to advance gene discovery in neurodegenerative diseases. And at the end, we will have Annabel Seyller, the Chief Executive Officer of the Annabel Open Science Institute, to give the closing remarks and maybe address any remaining questions that the audience may have. So with no further ado, I would like to welcome to the podium our first speaker, Professor Jason Karamchandani.
Good morning, everyone, and thank you. We're going to give you all a brief introduction into this platform, which I think is part of the fundamental, one of the driving platforms of open science here at the Neuro. And I like to start with this slide because I think it illustrates one of what motivated the creation of this platform. This is a peripheral blood film from a young patient who presented to an emergency room just outside of Austin, Texas. And 45 minutes after presenting to the emergency room, this slide was prepared and reviewed by a pathologist. And so within an hour, they were able to make a diagnosis of acute leukemia. In fact, just by looking at the cells, they were able to subtype this as an acute myeloid leukemia. And so this is a disease where the cells that drive the biology are extremely accessible. And one of the corollaries to that is that this is one of the greatest successes in oncology and medicine. The disease was given a name in the 1800s. The first chemotherapies were tried in patients in the 40s and 50s, but not a decade has passed without a significant contribution to how we care for these patients. And this slide's actually out of date. In the last decade, the invention of CAR T cells is yet another transformative addition to the clinician's armamentarium. And for some kinds of leukemia, ALL, the survival rate is now more than 90%. So in 1950, every patient dies. And in 2020, 90% of these patients. All right. So within 70 years, we go from a disease that kills everybody to a disease that we can handle actually quite well. And one of the reasons for that is because the cells are so accessible. You can take them, you can put them in a dish, you can try compounds, you can see how the cells respond. And if you contrast that to disease like Parkinson's disease, where we don't get our hands on the neurons that drive the pathology, we're in a different situation. And so the story in Parkinson's is not nearly as successful. This is an exaggeration, but it's true to say that we haven't had substantial additions to how we treat these patients over the last several decades. So in 2016, when the Neuro adopted open science, one of the guiding principles was the creation of an open science patient registry and repository that operates in an open capacity. And really one of the main goals is to allow our patients to participate in research and really empower them to be part of that journey. And this is a group that understands that there's a desperate need in neuroscience because many of these pathologies lack effective treatments. And within the next 2 decades, this is going to be financially one of the most significant areas of human disease in Canada. One of the reasons Stefano talked about how this is our beloved institution, and I think this is a really interesting building for this experiment to take place, because there's really about a century-long tradition of patients coming to the Neuro to receive clinical care, but also simultaneously participating in research. When people came here in the 30s for surgery, we were still mapping out the brain. So the consequences of these resections were essentially unknown. And it's really a partnership born of trust. So we're actually fairly lucky. We can include both adult and pediatric patients, patients who lack the ability to consent so that substitute decision makers can be obtained. We can use both pre-consent and post-consent, depending on the clinical situation, all with the goal of making sure patients really understand that the quality of their care will not be impacted by their decision to participate. We collect a wide range of specimens and materials, and what we collect will be affected by the patient's pathology. So as an example, we don't take brain tissue from patients who are not undergoing neurosurgery, but we can take a wide array of materials and in fact do so for several years now. Along with that, we collect a wide range of data, including clinical information, demographic information, imaging information, and Sally's going to tell you a lot more about the genetics, which we're in the process of integrating into one central database. In the next couple of months, we will link to the clinical PACS system so that all of our patients who have undergone clinical MR imaging, those images will be able to move in the research space to allow scientists to ask new questions. And that's quite exciting, particularly in those pathologies like multiple sclerosis, where almost every patient has had at least one scan. I think it's worth pointing out when Canadians do things well, I think that every patient in Quebec has a little dossier du santé du Québec, a little electronic medical record, and we are allowed to look through that EMR in order to populate our research database as well. And that's a unique thing here in Quebec. So this was the hope. The hope was that we would find a new way of looking at these materials and studying these diseases. So our patient partners come to the Neuro. We obtain different kinds of materials, including peripheral blood mononuclear cells. We work with Dr. Durcan, who will speak next, to develop these iPSC lines, some of which are CRISPR to develop isogenetic controls. And we end up with new patient-derived organoids to study these diseases and to perform drug discovery. And it worked. And these are images of patient-derived organoids, or affectionately called mini brains. The picture on the right is pigmented because these are dopaminergic neurons producing neuromelanin in a midbrain organoid. I'm a neuropathologist. I love looking at cells, and you can see that what these look like on a microscope recapitulate the developing central nervous system. So we've been doing this for several years now. We've collected more than 30,000 samples from well over 3,000 patients with relatively large cohorts in Parkinson's disease, which Dr. Stratton will talk about, ALS, neuromuscular disease, and a rapidly growing cohort in multiple sclerosis, with an excellent clinical team helping to drive that recruitment. We have developed well over 120 patient-derived cell lines with isogenic controls. And I think it's interesting that we work together in this building so that these materials go into Dr. Durcan's EDDU, and we sort of collaborate and work together to amplify the results of these collections. We're really the central site for a bunch of pan-Canadian initiatives, and that's in ALS and Parkinson's disease and neuromuscular disease and in autism and intellectual disability. And one of our hopes is that this is an accelerator for our young investigators like Dr. Farhan and Dr. Stratton, who will talk here today. All of the information is stored in a central database, and that includes an integrated laboratory information management system. So you can see what specimens are in the biobank at any time, and this is integrated directly with the clinical parameters, and we will be soon integrating the genetic results imminently. But this is what I really like. This is the kind of science that we can do because of this platform that we wouldn't be able to do otherwise. This is a fairly recent picture from a patient who came here to the MNI for a muscle biopsy. And those squiggly lines in blue are the neuromuscular junctions. And so we're participating in a project to study the neuromuscular junctions in patients with ALS, with the ultimate goal of having a clinical trial with a disease-modifying agent that actually helps remodel the neuromuscular junction. And our first patient came in for a non-invasive EMG last week, so we are actually recruiting patients with ALS to contribute to the pilot project to help drive this clinical trial. We've had to build some new tools because, to my knowledge, we're still the only patient registry repository working in the open space, and we developed open transfer agreements, manuscripts around digital consent codes. We had to build a novel governance structure. And coming in the next couple of months, we'll be launching our new virtual consent platform to allow us to recruit patients, not only here at the Neuro, but across the country and internationally. In the coming years, we will be partnering with other clinical groups, particularly epilepsy, neurocritical care, and neuro-oncology, as well as some research projects like the OmniBrain project being run out of the Brain Imaging Center. One interesting story that came in the last year was that there was a clinical trial here at the Neuro looking at a very specific genetic alteration in patients with ALS, ATXN repeat expansions. We looked at our cohort of more than 400 patients, and we found 4 patients with that genetic alteration. Due to screening criteria, inclusion criteria, only 1 patient was able to participate, but that patient is still currently in that clinical trial, which is ongoing. And so this has inspired us to sort of evolve this a little bit. We go from the initial vision to a broader vision, which Sally will tell you more about integrating the bioinformatics to really help provide even improved patient care using research sequencing and bioinformatics, and really to also help our patients understand the longitudinal nature of this partnership and provide them with resources to better participate in their own care. To date, we've completed 75 collaborations in the open space. So all the collaborations are use open science principles. The results of experiments are returned to CBIG, and that's true whether we're dealing with academic partners or industry. And this has really picked up in the last couple of years. We had about 3 collaborations in the first couple of years, but we now have well over 45. And I do want to note this is only possible because excellent partners here at the Neuro, and this is Dr. Genge in our clinical research unit, our scientific director, Dr. Fawn, and Alan Evans on the neuroinformatics side. And with that, I'd be happy to take any questions in the remaining 5 minutes.
So okay, so we have 2 microphones. So as we wait for questions that may come from our online viewers, I want to ask a question, Jason, which I think is in the mind of many listeners. So you mentioned collaboration with academia, not collaboration with industry. Can you elaborate a little bit on how industry perceives the open science approach to the Neuro?
That's a very interesting question, and one of the most interesting discoveries of this journey was when we initially brought up the concept of open science to our industry partners, we were met with some degree of apprehension. This is a new way of collaborating between academia and industry, whereas previously you provide a sample or a resource, you sign a transfer agreement, and that's the end of that collaboration. You never hear about the results. And so I think it was an interesting bit of education to help our industry colleagues understand the goal of the return of these results to CBIG after a mutually agreed upon embargo period, which for industry is only 6 months. But the correlate to that is our open transfer agreement is one of the most industry-friendly transfer agreements that I'm aware of. Because if you look at most university transfer agreements, there's all kinds of language around intellectual property, and the university retains its right to own part of whatever intellectual property is developed by the industry partners. That's actually really unfriendly to industry. And so our open transfer agreement strips that language out and is actually one of the most industry-friendly transfer agreements that I'm aware of today. So after we have these conversations, and we've had these conversations with large industry partners like Takeda, Merck, and Genentech, as well as small biotech startups, sometimes based here in Montreal and across the country. And after we get through that conversation, it's actually remarkable how positive those collaborations have been.
Thank you very much. Are there questions from the audience?
Thank you kindly.
Thank you very much. Okay, so now we need help to connect virtually with our next speaker. So I will let do that. Okay, wonderful. This is a presentation. So next is Professor Thomas Durcan, who is a fundamental scientist and who has led many very important efforts here based on the application of human-induced pluripotent sensitive technologies. And now we'll also speak in his capacity as director of the Early Drug Discovery Unit at the Neuro. So I will leave the phone on, who's joining us virtually today. Can you hear us now?
Yes, I can hear you, Stefano. Nice to see you. Good morning, Montreal. Good afternoon, Italy. Can you hear me okay and see my slides okay?
Yeah, I'm okay. We're okay here.
Sounds good. All right, so I'm delighted to be kind of talking today to everybody in this workshop and to actually kind of talk a little bit about some of the work we're doing at the Neuro's Early Drug Discovery Unit, kind of focusing on how we're opening up discovery through collaboration and outreach. I'm just going to move that over there. And so when we talk about collaboration and outreach, I guess the first thing I want to talk about is really this is a team effort. We're a team of over 40 staff trainees covering a kind of all manner of backgrounds from cell culture up to bioinformatics. And we've kind of really broken our group down into 4 main areas. At the core of everything we do, as Jason mentioned, is iPSCs, so induced pluripotent stem cells. And for these, we have a group to phenotype and CRISPR edit to make sure we have the highest quality cells for our work. This then feeds into our discovery assays and screens, into our 3D organoids and tissue engineering. And then a big part of the work we then do is on training and outreach to really build a kind of group of researchers on how to work with these cells themselves. And so this is really where it works. So Jason really kind of set it up nicely of how it all begins for us. It really begins at the patient level, with the patients coming through the neuro, with all the manner of these disorders from Parkinson's to ALS to MS. We're able to collect the blood, skin, and we're able to work with this then to reprogram it into an iPSC and then make it into any type of cell that we would want to work with from the brain itself, either 2D neurons, glial cells, and then more recently into kind of more complex 3D brain organoids or neurospheres. So we cover 3 main disease areas at the EDDU, Parkinson's, ALS, and then neurodevelopmental disorders, either intellectual disabilities and autism spectrum disorders. And we're really focused on making cells that really fit the disease itself or that are affected by the disease. So in terms of Parkinson's disease, this would be dopaminergic neurons. For ALS, for instance, it would be motor neurons. And then for developmental disorders, it would be cortical neurons. And we tried to match it with the 2D and the 3D. So to kind of have, if we have 2D dopaminergic neurons, then we would have a 3D midbrain organoids in which they're enriched with these dopaminergic neurons. Now, this kind of just shows you the full circle of how we go, and everything we do really relies on CBIG. Like they were this kind of thing that was set up before us, and because of them, it really feeds into everything we do because we're able to get patient material, consented in an ethical, open manner, and then we're able to then make them into these induced pluripotent stem cells, CRISPR edited if needed, and then this really feeds into an assay development and a disease modeling in a dish kind of model, either in 2D or within these brain organoids or bioreactors. And we are open science, and that's really what I'm going to talk about for the kind of few minutes I have today of how we feed into this open ecosystem of how we share our materials, our reagents, how we are able to share protocols, help training, and how to actually kind of do this without really going after IT in a way. So we set up in 2015. So we're still kind of new, although we're still a little old in COVID years now, 7 years. As I said, we do have 45 team members. We've trained nearly over 250 users in this time, and it's probably even a little bit more than that. We've got over 70 plus academic collaborations, and this grows on a week-to-week basis. And over the last kind of 2-3 years, we've put out over 35 plus open protocols and training videos to show people this is how we do it, so this is how you can then do it, so that there's no real trade secrets in terms of how we work with these cells, how we make the neurons, how we make the brain organoids. This has led to success in terms of metrics, if you want to kind of look at it that way. 30 plus open access peer publications, either those that we've published ourselves or in partnership with other groups. We've had over 15 plus industry partnerships from large pharma, such as Takeda and Merck, all the way down to small kind of startups, with Anuvio as an example. We'll talk about in a minute. We've generated a large catalog of iPSCs so we can get the cells from CBIG, and then these then go directly back into CBIG to feed the open science ecosystem. And then from the videos and the methods that we do, they've been viewed over 12,000 times, or for the videos we do in training, viewed over 4,000 times. And so really what our goal, or I guess our principles are for this, is how to open the potential of iPSCs for people, to make them accessible to all, really, to train users how to work with them and to train them how to work with them in a proper manner, and to really work with academics and industry so we can unlock the potential of these cells for discovery and translation with open science at the core. So when we talk about making our cells available, we have close to over 250 lines available at this point in time within 7 years, which when you compare it to other kind of repositories is very impressive. Every cell we have undergoes this very stringent quality control pathway where we monitor its cell growth, genomic stability, pluripotency. And if you get a line from us, you kind of get this data sheet where you know what it actually can do. And this then all can feed into the ability to be made into a cell type that you may want to work with. So Jason did kind of show you the numbers, or just gave you a number, and this is really what it is. We have over nearly close to 130 lines we've now made ourselves, and it grows every week to week. Sometimes it goes up by 2, 5, depending, and this is really kind of a very impressive team we have within the group. Covers Parkinson's, some individuals without any brain disease, ALS, neurodevelopmental disorders, and now we're growing and growing into rare disease and other kind of diseases as it really kind of comes on board and these initiatives launch within the neuro itself. And we've even been able to combine these with cells from outside. So in terms of Parkinson's, we've been able to get lines from the Parkinson's Progression Markers Initiative to target ALS or even Safari, for instance. So in total, we can offer close to 250 lines across any project to fit any needs of the different people that would want to work with them. And everything really for the lines we make in-house kind of undergo this really intense quality control validation. So if we either CRISPR a line where we might knock out a gene, maybe we repair a gene, this will then go into the quality control pathway. And if it's made from scratch, where we will program it, that goes into our pipeline after 3 passages. And then it undergoes STR profiling, make sure the cell is what it is, karyotyping, genome stability, ICC to make sure the iPSCs actually look like iPSC colonies, that they don't have any kind of non-differentiated cells. And some of the most kind of valuable stuff are the kind of the real thing to make sure the cells are good is weekly kind of mycoplasma testing and other testing. And then this all then either goes into CBIG, so that then it gets distributed across Canada, the world, whoever would like these to make the request, or we start to use it in our own projects, where we can then differentiate it into cell types of interest. And so we make these cells into neurons, astrocytes, any type of cell you may want. And then we then make the methods accessible. We have over 20 open access protocols. So these are written protocols. We've put them online. They're viewable on Zenodo. We have put a few online as pre-printed or actually accepted peer-reviewed papers as well, viewed over 12,000 times. And more recently, actually, thanks to funding from the Neuro Tannenbaum Open Science Institute, or TOSI, we've actually been able to get a little bit of funding to actually translate it from the written word into video protocols. We have about 10 plus video methods now available. These are available on our website for free, openly accessible, so that then when people come to us to ask, how can I work with a given cell line, then they can actually then see it within this link. And not just in English, we're actually making it available across different languages so that anyone in different countries can then see it. So we've gotten English, French, Portuguese, Spanish, and now we're starting to get into Arabic and Italian in due course, so that then people can then see it and then clearly understand what type of work we want to do, because we really want to open it up to the global audience. So to give you an example of a cell type we work with, so dopaminergic neurons, just to shows you the protocols we follow. This is the links you can find them at, and just to shows you the workflow we follow. It's adapted from kind of already published protocols online, where you can grow them anywhere from 1 week to 6 weeks and even longer, depending on the need of what you would want to do. And these are the cells, like from control lines or other cell lines, that we're able to apply across into different projects and collaborations. So one example I like to talk about is actually one of our earliest ones, where we worked with Dr. Francesca Cicchetti from the CHUC in Laval. We were able to make dopaminergic neurons with her and within our group, and to test their specific agent, histamine, kind of just as an early translational therapy. And we were able to show is that if you actually treated these neurons with OHDA, the neurons or the axons will very easily fragment. And then if you actually then added back in histamine, you could actually rescue this axonal degeneration. So it was kind of an example very early on to show we can make dopaminergic neurons of the highest quality, and they could actually be used to show results that were shown in mice could replicate in the human system. And now I believe this is kind of moving forward at this stage into early clinical program. Another example is it's not just kind of on the clinical side to test translational products. One of the other things is we want to actually understand basic mechanisms of science. And this is a collaboration we've actually had ongoing with Dr. Peter McPherson and his student Armin Bayadi for many years, where we really wanted to understand, how does synuclein actually get into cells? So synuclein is one of these kind of hallmarks of Parkinson's disease. Patients have these aggregates, but it's not really known well how synuclein gets taken up and trafficked through the neuronal network. Through work with Armin and ourselves, we were able to provide these fibrils, these aggregated form of synuclein, and we were able to show kind of this novel, never described before mechanism of synuclein getting in through macropinocytosis, combining it with high-end confocal imaging with this EM here. And this just shows you the synuclein aggregate here, right at the plasma membrane, getting taken up as the membrane kind of engulfs it, and then will pretty much traffic it to the actual lysosome very rapidly. And so this just then helps us understand these processes that happen, and then we can then start to kind of build kind of discovery or translational programs around it. And this is published recently in Cell Reports, and it's all open access, so anyone can then read the work that was being done. So we don't kind of just stop at 2D models. We also then go into more advanced models, so these 3D brain organoids that probably some of you have heard about. And this is where we would work from the human going into an iPSC, and then we divide them into a single cell mixture, embed them into this jelly-like matrigel substance, and then once we start to shake them, we can shake them and grow them anywhere from 2 weeks up to 18 months. And they grow in size over many different days. At day 1, they would be about 500 microns in size, and then by day 50, they'd be up to 4 millimeters in size. And the reason we like to work with them is they almost represent a very small piece of tissue for a different region within the brain. It gives you a kind of complexity to it. And we've been able to kind of use these across different projects and different collaborations. So one example was we didn't just kind of stick to working with people we knew. We actually crossed the street and started working with bioengineers, because we were noticing with these brain organoids, they're actually very soft, and the brain itself as a structure actually has a certain degree of stiffness to it. So this actually led to a collaboration about 5 years ago with myself and Chris Morais in bioengineering and a student, Camille, to see, could we take this brain organoid that we're making, which is a little squishy, and could we alter or adjust its stiffness to maybe make it a bit more brain-like? And so this was published about a year or so ago, and I think American Chemical Society, where we were able to show is by mixing in different percentages of alginate, you could actually modify the size of the organoid. You could actually make it stiffer, so that then when you would actually measure it, it would actually be a lot stiffer, a lot more compact, and then it would actually affect its overall structure. So through a collaboration where we would cross the street with an engineer, we were able to show that really we could then actually adjust and affect how an organoid could form and actually adjust its stiffness to maybe potentially make it more physiological. Another example of kind of work we've been doing is actually with a startup in Montreal Neurological Institute-Hospital. So what we wanted to do is we were making kind of brain organoids at a small scale level about 2 years ago. And we kind of went to them. This is really work by V, Magna, and Paula, where they kind of talked to Inovio to say, could there be a better way to do this? And this kind of led to the idea of these EB discs, or kind of that shorthand for embryoid body discs, where through these microfabricated discs, you could then start to scale up brain organoids into much larger numbers. This is what they now look like today. They're on the market. People can buy them. And once you can make them, you can really then you can actually then have these organoids growing for weeks to months, depending on when you need them. And you can grow them in bioreactors anywhere from 1 or 200 up to 800. And these can be used for many different processes. So making them is good, but then what do you want them for? I guess that's the question. And what we're really trying to apply these brain organoids towards is actually building patient profiles. The more patients that come in, the more iPSCs we make, then the more organoids we can kind of get generated. And we kind of apply different ways to do it. One is to see what do they look like, applying advanced imaging modalities, both either fixing it and taking very thin sections, all the way up to kind of 3D confocal imaging, where we're able to reconstruct it throughout. Or the other thing is we spend all this time making it to then actually start breaking it apart into almost like a cellular jigsaw. And the reason for this is if we can have it at its individual cellular components, we can then start to understand what's in it, how does it all, or how do these cells associate with each other, and if a disease is going on or a mutation, how does this impact on the different ratios of cells within this kind of organoid itself. And this is really work we're kind of starting to pioneer actually and build with Joe Stratton, who's been really kind of has come on board and really started to unlock and build the single cell group within the Neuro to kind of go from this single cell or this kind of brain organoid here into almost kind of a virtual map or an assembly of what the cells look like in these organoids. And between her and Sali, it's really given us the ability to do this at a much larger scale and to actually really build in the bioinformatics that is not something that I have even the background on, but thanks to them, I've really been able to advance it rapidly over the last 2 to 3 years. And so what really comes next then? So by opening up all of this stuff is from the patients, we can now get to their stem cells, screening small molecules, and even now, not just small molecules, there's a lot of biologics that are coming on board, from ASOs to other types of ways that kind of are being seen as potential new treatments. And then once we have these kind of compounds validated in a 2D human cell type linked to the brain, then the goal is then to validate this within a brain organoid, ultimately maybe more advanced abnormal models, and then eventually go into kind of clinical therapy. So this is really the 3D discovery pipeline we hope to follow over the next decade or so. And so this is really what we've been going at over the last 7 years, opening up the potential for iPSCs to make them accessible to all. If people are watching in Italy or beyond, we have a list online. We're happy to make them accessible, to link you in with CBIG and explain how to work with them, to provide you our training processes, to show you how to work with them, and to work with academics and industry across Canada and globally to really unlock the potential of these for discovery and translation, all with open science at its core. Because in a way, if we all kind of work on this jigsaw of the brain itself, we're really then able to make a lot more progress than if we work individually by ourselves. So I'm just going to leave here and pause, and then thank you for the invitation to talk today, and I'm happy then to take any questions.
Hi, Tom, can you hear me?
Yep.
Very, very good, thank you. So thank you very much, Tom, for the presentation. Before I ask a question on my own, maybe I want to ask if anybody here or online has any questions. So as questions may trickle down, so Tom, let's go back to again the important role of open science. Okay, so we all know, I shouldn't say that, but people who have been working with the models that you have described know how complex and laborious they are, and the need for many applications and so on. I'd like to hear your perspective on how you think open science in particular will help us address these enormous, massive efforts that we need to undertake, when studying many, many patients and lines and replicates from the lines and so on. So what is your thought about how open science really can contribute?
I think being part of the open science movement, I think the big part of it that we've learned is how to kind of make connections with people that we often don't have the skill set to really make a difference. So we can make the cells, we can make a lot of them, but then you get a lot of different data out of it. So brain imaging, so cell imaging data, there's electrical activity data now, and the single cell data. This all comes with new ways to do analysis, new ways to store the data, and then if you start to multiply it by 10, 20, 40, to really kind of have the informatics to actually parse through it, like in a way, instead of working on one cell, to work on 50 patient lines to get all this data, that requires a real team effort. If I was to do it, or my team, or two of us to do it, like as a standard project 10, 15 years ago, we'd be doing it for 100 years. Now, with the likes of Sali and Joe, where they have the kind of algorithms, the data management side there, we're really able to then both make it easier to adjust and analyze it in-house, and also make these data sets available so that we're able to access other people's data sets, and we're able to put our data sets online so that if people see the data, they can actually look at it, interrogate it, and maybe come up with new things that we're not even able to see. So I think that's going to be the big thing, is how to find all the right people to tackle these very complex questions in a way.
Thank you. Okay, we have a question, and so I will listen to your question, then I will repeat.
Okay. I'm curious how Tom communicates model neurodevelopmental traits in the dish. Things like autism, ADHD. It sounds a little crazy to think. What are you seeing? Are you seeing hyperactive cells? What are you looking for as a cell biologist when you are modeling neurodevelopmental psychiatric traits in a dish?
Yes, thank you very much. So the question is regarding neurodevelopmental deficiencies, psychiatric disorders. The question for Tom is, what is exactly that you can model in a dish that's relevant for this psychiatric and neurodevelopmental disorder?
Yeah, no, it's a great question. It's a very these are very complex diseases to model within a dish because it's such a nuanced disorder. I think we kind of are very focused on very simple cell biological traits almost in a way, how a neuronal progenitor may form an axon at the very beginning, maybe potentially its myelination, even how the different cells interact with each other. We have a number of projects looking on how astrocytes, oligodendrocytes, microglia, how they all kind of come together to form their relationship, and really kind of focusing on both the activity side of it and then potentially how they kind of connect and form. I think organoids kind of offers us another opportunity to potentially look at more advanced stages of the developmental process from 3 months up to 6 months, but it still kind of limits us in a way. We can ask kind of cell and activity-based questions and then bring in the transcriptomics, but I think this is where it has to be combined with the kind of the advanced animal models to then look more into, you know, the kind of cognitive side and some of that stuff that we don't or aren't able to get at.
Okay, another question from the audience, please.
I guess related to that, as we become more efficient at developing these models, from an ethics point of view, at what point do we stop? Are we making humans in a dish? Is there regulation around this in your field? How does that work?
So Tom, the question is, as we become better and better at making particularly three-dimensional brain organoids, at what time do ethical issues come into consideration? How are we going too far? Are we making mini brains? Nobody likes to call them mini brains, but we eventually going to make mini brains.
For me, I think there's still at an earlier stage. I think we do get advanced activity, but I think in terms of, is it where kind of you'd have a more sentient being? I think it's still at least a decade away. I know there are kind of ongoing efforts to actually start to build in ethical protocols for working with organoids. I think that's going to be coming online over the next few years. But I think the fact that a lot of them lack certain immune cells, vasculature, it's still only one piece of the tissue. I think it's going to be probably 5 years away. I think you're going to start to see it come in maybe in the middle of the decade as you start to get them more connected. I know there's more combined assembloid organoids. So I think over the next few years, you'll start to see more of that coming, but I don't think it's there yet.
Thank you.
Coming.
If there are no questions coming online, which I do not see, there may be one under here. Yes, okay, let me just check very quickly. Okay, no, this is it's something else. Okay, no, so I guess not. So thank you very much, Tom. We really appreciate that you were able to join us, even though you were not here today with us in person, but it's always good to see you.
Thanks, Stefano.
Thank you and all the best. And so now, really, we're going to, I have to be sure that Tom's presentation is closed or doesn't show anymore. I think we should be okay. So we're continuing, as Tom already alluded, by keeping a focus on the use of these systems to study neurodegenerative diseases like Parkinson's and applying more and more advanced technologies, including single cell and neurobiology. So it's my pleasure to introduce Professor Stratton, who will be our next speaker. She's also a fundamental scientist here at the Neuro.
Okay, thank you very much for the introduction, and really thanks so much for the invitation. I'm really excited to come after Jason and Tom because I actually have a presentation that talks about applying some of these open science resources that we have. The disclaimer now, this is heavier on the basic science mechanism side, but I'll try to keep it high level so everyone can understand. So as we've heard about, Parkinson's disease is a neurodegenerative disorder. And there's many mutations associated with this disease, such as PINK1, Parkin, LRRK2. We heard a little bit about synuclein as well. But it's important to note that these mutations alone don't drive disease. For the most part, there's also environmental factors. So it's a combination of genetic environmental interactions that leads to Parkinson's disease. And one of the defining features of Parkinson's disease from a pathological context is the loss of dopamine neurons. And you can see that nicely here in the substantia nigra, where you see this loss of pigment in the substantia nigra, which represents a loss of dopamine neurons. In addition, we know that there are many different types of symptoms in this disease. Motor symptoms is probably the most well characterized, but we have to keep in mind that early in prodromal stages of PD, before motor symptoms arise, there's evidence of some constipation, sleep disturbances, as well as depression and anxiety. So we're interested in understanding the entire spectrum of this disease as well. So what happens from an inflammatory perspective? I'm going to go through a few slides now showing what the evidence is in PD for having an immune component. So one, we know that inflammatory bowel disease increases the risk of Parkinson's disease, and this has been shown in several nationwide cohort studies. People who assess that fecal inflammation, we can see that calprotectin, which is a common marker for inflammatory bowel disease, is increased in Parkinson's disease. We're also aware of the Brack's hypothesis, which you may have heard of, that it's potentially a gastrointestinal, so intestines, colon, or olfactory initiating site that then results in propagation of pathology from these peripheral sites to the central nervous system via prions-like propagation, which then results in these loss of dopamine neurons that are in the substantia nigra. So if this is the case, we need to better understand these pathways as well. Another really interesting element is we see T cells, so immune cells are present in PD autopsies, interacting with neurons. So it might be that T cells are driving some of the neuron loss. And then ultimately, we also see an expansion of antigen-specific T cells in PD that's specific to Parkinson's disease. And when we put immune cells into a dish, you can actually identify exactly which peptides these immune cells from PD patients are responding to. So T cells expand in the presence, in this paper, of synuclein peptides, suggesting there is some sort of autoimmune response. So going a little deeper into autoimmunity, just to give you a bit more context, we know that neurons, if they're stimulated with stress or inflammation, can present antigens. And if T cells are present that are specific for these antigens, they can detect this antigen and bind to this antigen and then cause a directed autoimmune attack on the neuron, causing that neuron to die. So for this to happen in Parkinson's disease, you first have to have the T cells that are specific for the neuron antigen, as well as the neuron presenting those antigens. So we also have mouse studies that support this hypothesis. So when we inject or infect an animal in the gut, after several infections, we can see that a motor symptom arises in a pink-1 knockout animal. And this is reversible by L-DOPA. And when we look at this from an in vitro perspective, you can see that T cells are mediating this neuronal damage that's driving the motor symptoms. And L-DOPA reverses this, suggesting that dopamine neurons might be the neurons that are most affected. So since this pink-1 mouse study, we go in vitro with our collaborative team, and this is unpublished data by my colleague, Michel Desjardins, who's part of our PD immunology network here in Montreal. And he's been able to show that it's not just pink-1 mutations that are involved in dysregulation of antigen presentation. GBA, LRRK2, BPS35 are also potentially drivers. And because this is an in vitro model, we obviously need to take this back into a more complex system and confirm whether all these PD genes that are associated can indeed have a role in antigen presentation. So to summarize, personalized approach to PD would be very helpful. We know there's complex genetic environmental factors that drive disease and would be diverse among patients. And we also know that humans have unique MHC molecules due to a highly variable HLA alleles, which makes the process of antigen presentation highly individual as well. And so how do we determine the repertoire of antigens that might be presented in PD patients beyond, let's say, synuclein or pink-1 related? And so that's the work that I'm able to do in this context, having access to things like CBIG and EDDU. If we can define these antigen then maybe we can understand if there's a unifying T cell response across PD patients, define biomarkers in this context, and ultimately in the long run, there might be potential for therapeutic targeting of these antigen-specific T cells. But ultimately, one of the other major interests for me is whether or not we can detect these T cells early enough, or any immune response early enough in disease, before inevitable widespread neuron loss and dysfunction, which we know neurons, the irreversible damage and death is too late to really treat the disease at that stage. So can we find early novel biomarkers and perhaps treat patients before irreversible neuronal damage? So I'm going to talk a little bit about this first bit and then move on to the early detection part of my talk after that. So firstly, I have access, thanks to CBIG and EDDU, to PBMCs, mononuclear immune cells. And I currently have banked N equals 38 prodromal and established PD samples with healthy controls. And I also have matched iPSCs, matched to 4 of the patients in that immune cell analysis on the left. And importantly, I have HLA information. I mentioned before that HLAs are very variable across patients. And so if we want to be able to match any of our studies between immune cell and iPSC analysis, we need these genotypes. We need to know the HLA information. And one of the benefits of working here at the Neuro and CBIG is we already can get genetic information from these patients. Before I have to do any genotyping on these patients, in theory, I could get full genome-wide sequencing information, negating the need for me to complete my own genotyping. And so we've just done a small trial to compare my genotyping with the genome-wide sequencing data that's out there. And for the HLA, A, B, and C, we've got 100% crossover. So this tells me, Ziv Gan-Or, who does a lot of sequencing, gave me this data and tells me in future years, I can start to just get the genome-wide association study data and use that so I don't need to replicate, which is a really nice saving of resources. So when we take these iPSCs that I mentioned that are HLA standardized, I can create different immune cells and neurons. And this is an example of dopamine neuron generation that actually Tom already mentioned. It's very clear by 4 weeks post-induction, these neurons can start to express dopamine markers such as TH and DAT. So we use this system to try to show which antigens these dopamine neurons are presenting. So the first step is neurons don't traditionally express antigens at cell surface at baseline. So we needed to first create an induction system, which we use interferon gamma and LPS stimulation for MHC1 to be expressed at the cell or present at the cell surface. And what we've been able to show in dopamine neuron iPSCs for the first time is that we can get up to 90% MHC1 cell surface expression on dopamine neurons, which is quite high. And with this type of assay now, we can do immunopeptidomics. Immunopeptidomics is the method of taking the MHC peptide complex with the antigen that's being presented in that complex and then do proteomics on it. And by doing that, we've been able to isolate up to 6,000 peptides being presented by dopamine neurons. And that represents about 3,000 proteins in total. And we can look at what proteins are most highly represented. So there's about 120 peptides from the MAC-F1, which suggests that there are certain proteins that are more likely to be presented. And then we can do prediction based off of those proteins as to which ones might be most immunogenic, which are most likely to elicit a T cell response, which, there's going to be so many peptides that we do need to somehow reduce the number we're working with. And so it's a system like this that's made available due to the CBIG and EDDU platforms that will now allow us to validate whether any of these peptides are of relevance to PD. So first, we've developed an assay system for discovering antigens using human dopamine neurons. And now we're determining whether these highly ranked peptides can elicit T cell responses using peptide stimulation assays. So we thaw out the PBMCs from our patients, and then we add peptides of interest to see whether we can see clonal expansion of the T cell population. And if we're able to see that, then naturally, we want to co-culture these PBMCs with the iPSC neurons to see if we can get direct attack of the neuron, which would really prove the hypothesis that there is autoimmunity in PD and allow us to learn about the diversity of antigens that might be involved in this autoimmunity. So that's it for part one, where I really leverage EDDU and CBIG resources. And now I'm going to go on to a little bit of my mouse work, which complements some of this human work. So I mentioned before that we have a really nice PINK1 model where at late stages, we see motor phenotypes in response to gut infection. And so the PINK1 knockout seems to be essential for this. And what we don't know is, can we recapitulate the earliest stages of disease that we know is present in PD? So is there constipation? Is the gut hit first, like the BRAC hypothesis would suggest? And are there inflammatory mediators that could be early biomarkers of disease before we have late stage motor symptoms? So the first data that we were excited to see is by 2-week post-infection, which is about when the infection is being cleared, we have a reduction in fecal water content, which suggests a constipation phenotype. At the same time point, we also see less number of fecal pellets being produced in 1 hour, also suggesting that there is a constipation phenotype. So what is underlying this constipation? We perform single cell sequencing, assuming that it might be the immune system, given we know that inflammation is associated with PD in the gut. And what we found is there's one cell type of particular interest at these earliest stages of PD in this model, monocytes that seem to have a large number of differentially expressed genes. And so at 1 week post-infection, we began to analyze this data set using big data bioinformatics just on the monocyte population. And so we started by doing a Go term analysis, and we found that several interesting Go terms came up, like pro-inflammatory regulation and IL-1 beta signaling, in addition to several others. So we started to follow up on these two particular observations, and we did a little bit more bioinformatics to see how IL-1 beta was involved. And you could see on the top here that there were several IL-1 beta response genes being upregulated in monocytes. And when we look at one in particular, lipocalin, it's clearly, when we performed ELISA in the gut at 2 weeks post-injury, also upregulated. So we think IL-1 beta might be involved in driving the pro-inflammatory response, lipocalin being one of a major mediator in pro-inflammatory mechanisms. So looking at more of these IL-1 beta response genes, you can see there's Go terms relating to neuron apoptosis, regulation of inflammation, reactive oxygen species. So we wanted to follow up to see whether monocytes have a pro-inflammatory phenotype. And we wanted to do this in an independent system, not in vivo. And so we took bone marrow from these same animals, PINK1 or wild type, and then we stimulated these cells after differentiation into the myeloid lineage. And what we could see is IL-1 beta LPS stimulation actually had the most number of iNOS positive pro-inflammatory myeloid cells in this context. So we were interested to know if it's these pro-inflammatory cells that might be driving some sort of neuronal damage early in the gut that could be underlying the constipation that we saw. We know constipation is driven by a lack of peristalsis, movement of gut motility, and if neurons aren't innervating the gut properly, this would result in this constipation phenotype. So the next step was to create a primary enteric neuron culture. So we isolated neurons from the gut of these animals. We also had a nice DAP-CRE reporter, which allows us to monitor dopamine neurons in the gut. And then we added conditioned media from these myeloid cell cultures that I mentioned before, and added the conditioned media to these enteric neuron cultures. And then we used PINK1 neurons as our base, and then we added conditioned media either from PINK1 knockout or wild type myeloid cells. And what we were pleased to see is when we added the myeloid cells that were stimulated with conditioned media from LPS and IL-1 beta stimulated cells, there was clear reduction in the number of TUJ1 positive neurons. So there was a reduction in neurons in our culture dishes in one condition that was dependent on PINK1 knockout. And when we looked at this in the DAT positive population, so the dopamine neurons present in the gut, we also saw the same effect. So it seems like dopamine neurons are affected very early in disease, potentially partly underlying the constipation phenotype that we're seeing. So to conclude this part, it looks as if we have a dysregulated innate immune response that might be driven by the myeloid cell population early in disease. And it could be that at this early stage already, the neurons are being affected in the gut, which could be driving constipation. Now, what we don't know is how this kind of early response, one, can be used as a biomarker that we might be able to learn from to better diagnose disease early. And two, how does this early dysregulated inflammatory response then relate into a late stage autoimmune response where T cells are driving central nervous system neuronal loss? And we have a little bit of evidence to suggest B cells might be involved. So at this early stage, XPP1 is upregulated on a gene transcription level only in the knockout infected group compared to the other experimental conditions, suggesting that there's more antigen presentation even in the B cell population. So we're trying to put it all together at the moment to look at the myeloid cell populations, B cell and T cell populations to try to understand the progression of disease now that we have this really nice time course model that models the natural evolution of disease. So with that, I have to thank the group. It's Sherilyn Racinto and Adam McDonald here on the slide have been absolutely amazing with driving both the human in vitro assays as well as the animal modeling. And I have to also thank the PD collaborators. I'm new to Parkinson's disease research, and really the group at ASAP has been critical for me to learn everything and get moving on this project. And of course, Tom and Jason, who spoke earlier, for their valuable input for CVIG and EDDU as well. So with that, I'd like to take questions. Thanks.
Thank you very much. So I have to use the same microphone to be sure our virtual participants can hear us. Any questions from the audience? So I do have one question. So it's clear that from the in vivo work with animals, you can identify important responses or changes in enteric dopaminergic neurons. So if you want to then model this in humans, what is the current state of knowledge in terms of deriving enteric from iPSCs?
So it's a very, very good question. Actually, someone in Tom's lab is working on that. We have another collaboration looking at protocol development for enteric neurons. So Sherilyn, the student I mentioned on the last slide, is working with the EDDU to try to develop the protocol. There is one published protocol, but there's been a lot of playing around internally to try to get it going. But I don't think they would include the dopamine neurons. So that's something that we're still trying to assess. If we could bring this back in vitro, would be absolutely amazing in the human system. Yeah.
Okay. Yes. Okay. Yes. So please go ahead, and I will repeat that. I will then repeat your question.
Thank you, Dr. Great talk. I was wondering what you think we are students that really why it's just targeting just like the dopamine amounts of the brain and why we have to see, similar movements for where we are.
Yeah.
Maybe you want to repeat the question?
Yeah, sure. Yeah. So the question is, if it is an autoimmune disease, why doesn't it pathologically in the brain look a little bit more like MS with more immune cell infiltration? That's a very good question. And I guess based on my work, the hypothesis would be that dopamine neurons present a certain antigen profile that other neurons don't. And that's why we're spending the time to differentiate these iPSCs into dopamine neurons and then doing the immunopeptidomics. If there's a specific profile associated with the subtype that's dependent, then it might be the difference. But this Brax hypothesis where you see like progressive pathology, it's so different to what you see with the MS pathology. ALS is similar in this kind of prions-like progression, and there is evidence in ALS as well, looking at autoimmunity and how T cells are involved. So I don't think we quite get why, but we know that it's a common theme across neurodegeneration that there's suggestions of autoimmunity. And even Alzheimer's disease has some genes associated with immune dysregulation. Yeah.
So I'm wondering how you think maybe like with the prion pathology, those specific neurons are calling themselves to dispose of them because
yeah.
as opposed to something more like
yeah. Yeah, I mean, dopamine. Oh, yeah, sorry. Okay. So is it something about these particular neuronal subtypes that might be calling the immune cells there in order to drive it? That makes perfect sense. Actually, there's another PI here, JF Poulin, who's just got a CHR relating to what is the immune signature of neurons and what's the interaction that might be targeting these cells from an immune perspective. So it's true. It could be something as simple as that. We know dopamine neurons are generally more susceptible to injury and damage given certain features of these cells. So you're right. If these cells just simply express more antigens, they might be getting targeted more. And then the microenvironment, the microglia around them, creates perhaps also an extra added stress that causes the damage to these neurons. Yeah.
Okay, thank you so much. We will move on for sake of time. So last but not least, the pleasure of inviting to the podium Professor Sali Farhan, who is also an integral member of our open science community. And I think she will bring a bioinformatics perspective now, in addition to what we heard before. So welcome to the podium.
Very much. Oh, I have to stop sharing.
Yes.
Sorry.
No?
Oh,
Okay. Yes.
yeah.
That's good. Just
Okay.
I hope looks good.
Okay. Okay. Somehow I figured Google Cloud, but still focusing on Zoom. So good morning. Ciao. Bonjour. Many thanks for having me. My name is Sali Farhan, as Stefano mentioned. And today I'll be talking about how we've leveraged open science resources to advance gene discovery in neurodegenerative disorders. So I'm very lucky to be at a place, the Neuro here, where we study many different types of neurological traits. This is specifically neurodegenerative slide here. This is an iconic slide that's been shared many times as well. So many of these disorders we do study in-house as well. And these are separate clinical entities. Some of them we know them quite intimately as well, and then some of them are less studied given how rare they are. But really they are separate clinical entities. They affect different regions of the brain. And as our clinical colleagues will know as well, some patients can present with single neurodegenerative disorders, as well as they could present with multiple types. But really the applications that I will be discussing today can be applied to any neurodegenerative diseases, any disease in particular. Okay, so this is another iconic slide in my field. I'm a geneticist, so I study the genomic signature of amyotrophic lateral sclerosis, or ALS, as well as a few other rare neurological disorders. So this is a fundamental slide that we use when we're teaching graduate students, undergraduates, and postdocs as well. And even though it's so nicely presented here, it's years of development, years of methods application that was shared, whether through publications or open resources, time and time again. And these same principles developed by geneticists are applicable to all diseases, as I'll discuss as well. And so we've been able to discover many different genetic profiles of all neurodegenerative diseases as well. So on the x-axis here is how rare a specific mutation or a variant is in the general population. And typically we see this very close relationship with how rare a specific mutation is and its effect size, its biological impact on disease. So there are diseases that certainly can present as both simple. A simple mutation indeed results in the phenotype. Then there are more complex traits that we study here at the Neuro, as well as other neuropsychiatric traits like schizophrenia, or even something as common and just a simple trait like height can actually be quite complex. Multiple independent mutations each gather together and produce a trait, a phenotype that can result into either a disease itself or something as simple as our facial features or personality, etc. So these applications, again, have been used across all diseases. So the disease I study is ALS, and this is quite a severe filial disease. It's characterized by degeneration of motor neurons, leading to progressive weakening of limb, bulbar, and respiratory muscles. This is a death sentence to these patients, so urgent need is absolutely needed, as my colleagues would know. So unfortunately, the majority of the patients diagnosed with ALS, they do develop respiratory paralysis, and within 3 to 5 years, this results in their death. There is currently no effective cure, but there's a lot of considerable effort by our industry colleagues, our academic colleagues, working towards approving and regulating therapeutic options for these patients. And I'd also like to highlight as well, this may have already been taking off, certainly in Europe, in more of progressive social countries like Belgium. But this specific disease was actually one of the main diseases that overturned the Supreme Court of Canada ruling in favor of legalizing doctor-assisted suicide, or MAID, in 2016 as a nation, and even earlier actually in Quebec. So the genetics of ALS, as I alluded to earlier, it's both simple and complex. So this is a tough one. We certainly have genes that once mutated, they result in disease, and that's the end of that. And then we have forms where a patient presents and we really can't find a single gene causing their disorder. We find risk factors, we find even risk activity, but we can't exactly identify what has caused their disease. So incorrectly in the field, we call some of these simple cases familial if they simply have a family history. And this actually led to a bias in our field in that we over-focused on these more simpler forms of the disease. We found genes, which is wonderful. We noticed that these genes can also be mutated in the more complex forms as well. But there was a bias to study more of the simple form of this disease. And then it led us to abandon more of the complex forms. And this has changed through collaboration, as I'll talk about in my upcoming slides as well. So how do we discover genes, or ALS genes in this context, but genes in general? Well, it depends on their effect size. Where there's smoke, there's fire, so you know exactly where to look for that fire. And this can be easily identified in linkage studies or familial studies where mom had the disease, dad had the disease, and then the grandchild eventually developed the disease as well. And we've seen a lot of success in this specific area. So we've had a lot of genes discovered through this matter, not just in ALS, but in multiple other disorders. We had this candidate gene study. So if you're a statistical geneticist like me, you would not be in favor of this approach because it's quite biased. It hardly ever replicates. But nevertheless, this was what we had at the time. And to give credit where credit is due, when investigating a particular pathway compromised in disease, certainly we were able to make inferences about other genes that were independently replicated. And then there's the large-scale, statistically rigorous genetic associations. And there's been many examples of ALS genes discovered in this method. So this is the least biased, but you need a lot of resources to conduct a study like this. You need many, many samples and many controls as well. So when statistical geneticists talk about power, et cetera, they're really focusing on identifying something that can't be picked up in more of a smaller pedigree, for example, as well. But I always want to emphasize that these are not independent. One must ensure that one method can certainly replicate another method as well. So you may have indeed found a bona fide ALS gene as well, but does it come up in families? Does it make sense in the biology, etc.? So we want to make sure that any gene that we do discover follows this simple pathway as well. So I always get this question too from non-geneticists is, okay, well, have we figured it all out? We've discovered so many. Are we going to stop looking into the genome? And I say yes or no. And so I've learned a lot from cancer and how it's not just that you're looking for a new gene. It's not just that you're looking for a genomic signature, but you're expanding the pathways that we already know are compromised in diseases. You're learning whether a genomic profile is associated with a specific type of subcategory of the disease, and you're moving forward accordingly. So this is just an x-axis showing the timeline. So the first gene, SOD1 was discovered, and actually our director was one of the authors here, as well as a few other OGs in the field. So it's kind of nice to see it still here. And then, of course, with time, with the advancement of technology, we've seen a considerable increase in gene discovery as well. And so the different color represent the different broad cellular function of each gene. And then the different shape represents the methodology as well. And so not surprisingly, given the evolution of technology, we've seen more of these statistically rigorous next generation sequencing applications lead to gene discovery as well. And then, you know, each method has its strengths and weaknesses as well. So some methods, it's kind of like obtaining legal advice from a Harvard law school graduate, and then some methods, it's kind of like obtaining legal advice from somebody on Facebook or something like that. So certainly some methods have stronger and more reliable results than others as well. But the goal is to always replicate what we find. Moreover, though, more targets can be more problematic as well. So this is a review from the different neurological disorders and how well they've advanced to clinical trials as well. So, you know, something we always hear about is that over 80% of clinical trials fail. Well, what's going on there? Is it simply the recruitment? Did we have too many targets? What exactly is the issue? So typically, disorders in which they have one specific target, okay? So one gene that's mutated that causes the majority of the disease can really advance to human clinical trials much faster than more of the complex traits as well. And there are many reasons for that as well, but you know, a simple analogy would be the classical coyote roadrunner analogy, in which you have one specific target, you can really study it really well and try to resolve what's happening there. But in some of the more complex traits that we have that are not necessarily caused by one single gene, you have multiple roadrunners. And this could be very tough for a single drug to try to get all of the roadrunners. So this is one busy coyote, and it may lead to a failure or halting of clinical trial as well. So I'm going to briefly mention an opportunity where we all came together as an ALS community and collected as many ALS samples as possible across the world, both cases and controls, and we were able to discover a novel gene. And this would have not been done had scientists not been willing to share their data as well. And so I won't go into too much detail, but this was kind of my entire postdoc. So it looks pretty nice on one slide. I don't know why it took me so long. But as a geneticist, we did a lot of the gene discovery, and a lot of information and a lot of quality control goes into this as well. So I don't do it justice by just highlighting it so rapidly here as well. But what we were able to do was collect as many ALS cases and controls as possible without overuse of resources. So these were previously collected cases, and almost every ALS investigator who had cases were willing to share it as well. And in doing so as well, it was the largest exome-based case control study in which we replicated many of the known ALS genes and then found a new DNA gene called DNAJC7, which fit really well in heat shock protein. We identified many distinct mutations in patients and took them through the route of ensuring and kind of validating that ALS patients known to carry this specific type of mutation were indeed had cells that actually showed decreased expression of the specific DNAJC7 gene. We also were able to expand this, so we connected with a heat shock protein yeast biologist to try and understand what is happening in the disease state. It's not happening in the state as well. So this really was an example of using existing resources, new technology, new applications and methodology, and new gene discovery, and then also taking it all the way to understanding biology as well. I want to thank all my collaborators and colleagues for helping us with this discovery. I want to also emphasize that all the data generated from here is made publicly available. So this is the ALS Knowledge Portal. This became the Neurodegenerative Disease Knowledge Portal, and it's fully publicly available. So anybody across the world can access it. And so this was actually in close partnership with my colleagues at the Broad Institute, Jason Flanick, and my colleague Mike Knowles and Andy Singleton at the Center for Alzheimer's Disease and Related Dementias at the NIH. And so within open science as well, one has to be very careful in not reinventing the wheel, which happens quite often. So Jason Flanick, hats off to him, he had developed this entire ecosystem of knowledge portals. So he studies type 2 diabetes, and he's made just such an elaborate resource in the type 2 diabetes community. He was also able to leverage the type 2 diabetes knowledge portal and apply it to musculoskeletal disorders, cerebrovascular disorders, and among a few others. And so I was just a postdoc, and I really wanted to create something like this for ALS. And so they kindly were able to offer me their resources, actually at no charge, and created the ALS Knowledge Portal for me. And then I really wanted to ensure that other disorders where genetic information can also be deposited into the Neurodegenerative Disease Knowledge Portal, and they repurposed it and into the Neurodegenerative Disease Portal, and now this is publicly available. And so as I alluded to as well, as our clinical colleagues would confirm, so some patients certainly just present with ALS, but some may have some forms of Parkinson's or frontotemporal dementia. So now a researcher can look at different phenotypes and see if one gene that was previously thought to only be implicated in ALS could also be implicated in these other disorders as well. So we continue to expand this resource. And I really want to thank them for helping us advance this community as well. Okay, so I just wanted to highlight our use of publicly available resources as well to advance ALS genetics. And so not all genes, not all ALS genes and their variants exert the same impact as I mentioned earlier. So actually we are a part of a global effort. It's called the National Institutes of Health in the States, but it's a global effort. So it's the ALS Clinical Genome Resource. And what we're doing is we're actually resolving which genes, as I showed you previously, are truly causal of this disease versus risk genes or even false positives, because the method that was used may have not been the most rigorous as well. And so we've led a paper that's coming out soon as well, where we've taken out all the ALS genes or posed ALS genes and tried to determine how many variants within these genes are truly causal of disease. So you can see that some of them have red, which is quote unquote bad, and there's a very specific criteria that a geneticist would follow to classify such variants. But you can see almost all of them have this blue, and what this blue is, it's uncertain significance. We just don't know. Are these mutations causative? Is this gene itself causative, or is it not? And so what we're doing is we're trying to work as a community to say, no, stop, this is not a correct ALS gene, or the variants itself among those that are blue, among those that are uncertain significance, what exactly is happening? And so this actually has immediate clinical implications because there are patients that get genetic testing, as Jason had mentioned earlier. So depending on a patient's genomic profile, they may even be eligible for clinical trials and even possible therapeutic intervention. So all of our efforts, as I described to as a part of this group, has immediate clinical implications, and we see that right here in Quebec. So I'm very proud as we now have ALS testing available for patients. So these are the genes that are more likely to cause the disease. And in the event that they are negative, but the clinician feels strongly that this is a genetic form of the disease, they have the option to have exome sequencing with what's called a virtual panel, where we interrogate the ALS genes first and try to see if we're able to capture the genetic cause of the patient's disease. So this was actually an effort that I led with our director, Guy Rouleau, and now it's available now in Quebec. And so our goal is to also make this available to all ALS patients in Canada and even globally if need be, although there are other commercial labs that offer this as well. And as Jason mentioned as well, a specific genomic profile can lead to a clinical trial or even therapeutic intervention. We see this happening in the cancer field very well. So we're using these successful examples and trying to reapply them. So we do have clinical trials here led by Angela Genge, the clinical research unit at the Neuro. So in the event that we identify any patient that meets these criteria, we're able to connect them with her and her team. So this is great. The other application that we've been using to understand ALS biology as well, the applications of AlphaFold protein structure. And so AlphaFold recently came out, and it's an artificial system, artificial intelligence system developed by DeepMind, and it's focusing on predicting 3D protein structure from the amino acid. And so all of those variants of uncertain significance I alluded to earlier, we're really trying to make sense of them. And so this is my colleague, Samaya Iqbal from the Broad Institute. And so sometimes we think about open science as in sharing data, sharing code, sharing resources. But one of the best resources is actually the people who have already developed something so elegant, so useful in another field. So she was able to show this in neurodevelopmental disorders. And I saw her lecture and I thought, wow, we could really use this in ALS. So hats off to her for coming along and working with us to try to understand of the variants that we've identified in ALS, how can we get to know them a little bit more at the proteomics level and make some conclusions from them as well. So what we ended up doing is taking all of those variants of uncertain significance, mapping them onto AlphaFolds, using very rigorous quality metrics to study them further. And so these are kind of some of the questions that we had asked. I won't go into too much detail, but we really interrogated all protein characteristics. Which amino acids are frequently mutated? Which secondary structure? What type of accessible surface area? Functional features, et cetera, et cetera. The high or low expression important. Post-translational modification important, yes or no? So we did a lot of these bioinformatics studies to try to understand and characterize these variants of uncertain significance in ALS. And we're working on finishing up that manuscript soon as well. And so a gene that we all agree is indeed an ALS gene, but we don't know if all of the variants harbored within that gene do cause ALS. So we're modeling them using the successful system that Samaya had built and reapplying it to ALS as well. So these variants that we don't exactly know what we're doing, we're able to make some inferences based on their protein characteristics and determine whether they are actually causal of a patient's disease or not. So with that, I just wanted to summarize our mission and some of the work that we're doing here as well that complements and advances open science. So we share all our in-house data sets with the academic community. I want to thank Jason again for inviting me to join his mission. And I'm happy that to share that we've set up some SOPs and workflows to make sure that every genetic data produced at the Neuro will be publicly shared with the world. As well, we want to bring in as many publicly available data sets into our own institution and advance our own science. So that way we're not just regenerating resources, as Joe mentioned as well, we're actually leveraging what's already out there to advance our science. And I mentioned to you before, we're leveraging these, again, publicly available resources, whether that's software, data sets, and even collaborators. So my collaborators, Jason Flanick and Samaya, they were both very kind enough to work with us to advance our own science and apply what was successful in the type 2 diabetes field and the neurodevelopmental field in ALS specifically. We collaborate closely with local scientists and made ALS genetic testing available in Quebec, as well as global initiatives dedicated to resolving the genomics of neurodegeneration. And again, we're sharing everything we produce, whether that's through preprints, our GitHub code. We have a neurobioinformatics core here as well, where we share all of our code publicly. And we're happy to participate and collaborate with everybody, whether that's here in Montreal and in Italy or all over the world as well. So with that, thank you very much for inviting me, and I'm happy to take any questions.
Grazie a te. Italian is becoming popular here. So, any questions? Okay, but then I have one. So, and again, it's kind of a conclusion to our 4 presentations. So we heard a lot about the wonderful ways by which open science can promote a number of steps, patient recruitment and empowerment, development of new models. But then you touched on something that I thought was very important. Arriving at the final frontier, the clinical trials. So how do you see open science contributing to that particular moment? Let's say the development of effective ASO agents or other agents. How do you see open science acting there?
Right. Yeah, so, as Jason talked about as well, the CRU team here is integral in that. So that's wonderful. And they were able to kind of tell us, these are the different clinical trials available here at the Neuro. So with that being said, then we're able to screen and flag certain patients with a profile. And we're able to either redirect them unless they've already know about them as well. So the best effort is knowledge sharing, what's happening, knowing what's happening in your backyard, as well as across the world as best as you can. I'm a part of another NIH group wanting to focus on the top priorities in ALS. And a colleague who's very famous in the field in Florida was mentioning he was looking for a specific type of patients who have a profile. And I thought, oh gosh, Glenn, we're fully open science at the Neuro. We could share this information, but I didn't even know that you needed it. So I think just communication, being very transparent with what we're working on, and trusting each other and sharing our resources, I think would probably be the best way. And just making sure that we do our best, be the best version of ourselves as scientists, as clinicians, to make sure that we share our knowledge and that everybody is aware of the resources that we have. Which is a bit more philosophical, but.
So thank you. So we have one question from the audience. Maybe you will repeat the question.
Absolutely, yeah.
It's fascinating what open science can bring, that we're finding all these associations that we didn't realize existed. When you add them all up, how many are left ALS patients, like percent-wise, that don't have an association that might be truly idiopathic?
Yeah, that's oh yes, absolutely. Sorry. So we're advancing science, we're advancing gene discovery, but of the patient population, how many of them have an idiopathic and unknown or truly non-genetic form of the disorder? And probably a lot. So, I think it would be naive to think that every disease, certainly ALS, every ALS case can be attributed to a genetic perturbation. With that being said, though, there is an interesting idea. So we certainly have a genetic risk for everything. And I say risk, it's not risk, it's just, the lottery. So for example, a trait like height, up to 80% of height can be explained by genetic variation. So how tall your parents were, but then in addition to that, 20% is left for so many things. So that's something like height, which is a highly heritable trait. So something more complex like ALS, like other, IQ, etc. There's certainly a strong genetic component, but we don't know everything, and it would be naive to think that we could explain everything. With that being said, though, certain fields like cancer have been able to be so much further ahead of us, as Jason talked about as well. So what can we do in these fields, like type 2 diabetes, like cancer, that have interrogated and used almost every technology under the sun? So how can we bring that to more understudied rare neurological disorders so that I can confidently answer your question and say, well, we've assessed, the 20, 100 methods, and the conclusion is that X percentage of patients cannot be explained by genetic perturbations. So stay tuned, I guess.
Okay, thank you very much. So we have about 10 minutes left, so we are right on time. And before we close, I want to thank the speakers today. I think that they presented to you really, really nice examples of our efforts, our local efforts to implement open science in our daily life. But what I want to say also is that, to build something that truly promotes open science within our walls and beyond our walls in ways that I hope you've appreciated, it takes brains and it takes muscle and it takes effort. And I would like to introduce now what I consider the brains and the muscle behind our open science initiatives at the Neuro, the CEO of TOSI, the Annabel Open Science Institute, Annabel Seyller, who will close this workshop with a few remarks overall.
Thank you, Stefano. Hello, everyone. Good to see you today. I would like first to thank our speakers, our wonderful hosts, Dr. Stifani, and all of you who attended, both in person, but also remotely. A very, very special, heartfelt thank you to the Fondazione Prada, who gave us a very unique opportunity to present to you today how we at the Neuro are actually putting open science in practice. When we embarked on that journey, on that adventure a few years ago, like Dr. Stifani was mentioning, it was crystal clear that open science was not the end goal. Open science was a means to a higher end, which is basically a mission enabler for us, impacting people's lives. The Tannenbaum Open Science Institute's first goal was to crystallize practice of open science here at the Neuro. Listening to my colleagues today, I think we can be proud of what has been accomplished so far. I'm particularly impressed and grateful to my fellow colleagues, that are still in their very early stages of their career. So I want to say a big kudos to all of you, Sali, Joanne, Jason, and Tom, for your dedication and commitment to science done right. It means a lot for us at the Neuro, and we really appreciate your efforts. I would like also to emphasize the unique contributions of the partners and unique partners of the Neuro, which are patients and their families. You saw today a unique ecosystem of platforms, of projects, of initiatives, but that would not be even feasible and possible without patients' contributions and family commitment and collaboration with us. Like Dr. Stifani was mentioning, we also created TOSI to incentivize other institutions across the country to embark on open science. I can happily report that we have recently been joined by two other large organizations in Canada, the Douglas Research Center and the Hotchkiss Brain Institutes, and many others are in close discussions with us as we speak. So we feel that momentum for open science is right and is coming here in Canada. But practice of open science needs to spread all across the globe, not only in Canada. And we feel, that momentum is also starting to feel right in Switzerland, in Paris, in many other countries. And large visionary organizations like the Fondation Prada and many others do have a very important role to play in crystallizing and triggering that culture change. I wish all participants of the Human's Brain Conference in beautiful Milan a very fruitful conversation with hopefully many, many discussions towards open science. I wish you all a very lovely rest of the day. Thank you.
Thank you very much. This will end our proceedings today. So thanks everyone. And I message to our Milan colleagues that we are now signing off. Thank you. Bye-bye.